thank you, Dave, for that introduction, and it certainly is a pleasure to be here this afternoon and share with you some of our interests and our program here on functional foods for health, and I'm going to be talking more specifically about our Ohio State University program. So let me just start out by defining what are functional foods. And this concept actually got started back in Japan many years ago. But functional foods are defined as food or food components when consumed as part of a typical diet, actually provide a health benefit that goes beyond just basic nutrition. Quite a bit of interest, of course, in functional foods by consumers. They drive a market, functional food market, that's estimated at about $38 billion per year and still growing. Healthcare professionals have an interest and understand, of course, that many diseases seem to be diet-related, and hopefully functional foods may prevent chronic diseases. And from a public health perspective, to address nutrient deficiencies and longer term reduce healthcare costs. So we're very fortunate here at Ohio State to have the Center for Advanced Functional Foods Research and Entrepreneurship, and as Dave mentioned, this got started with support from the Ag Biosciences Innovation Grant Program. It's a collaborative, multidisciplinary functional foods center. I helped direct the program with my good colleague Steve Clinton from the medical school. Mark Faella was initially involved for many years from education in human ecology, from human nutrition, and we're very fortunate to have Robin Ralston, our program manager, helps us operate this center program. So I think one of the greatest advantages of having such a program here on campus is it really enhances collaboration between investigators from diverse disciplines, horticulture, crop science, food science, nutrition, pharmacy, medicine, and public health. What we do is we develop some unique nutritionally enhanced functional food products targeted towards disease prevention and control. We have a particular emphasis in cancer prevention, as you'll see in just a bit. Conduct research studying safety, efficacy of these functional food products. We do our best then to try to disseminate some sound scientific information on the results of our studies, and also collaborate with industry, food industry, and business partners that have an interest in perhaps studying efficacy of their products and bringing them to the marketplace. So our collaborations cross a number of college boundaries here on this campus, from arts and sciences, education and human ecology, where human nutrition, human sciences is located. Of course, our college is heavily involved. College of Medicine, we have a strong relationship with the Comprehensive Cancer Center, the Molecular Carcinogenesis and Chemoprevention Program. We work with a number of investigators from different departments and divisions in the College of Medicine, also College of Public Health, Pharmacy, Veterinary Medicine, more recently with the Food Innovation Center, and also the Glenn School of Public Affairs. So we have this theme within this center program we call Crops to the Clinic. And we target a number of different areas, horticultural sciences, plant science studies, enhancing nutrients, phytochemicals, bioactives, and also the sensory properties of some of the ingredients in the crops. In food science, we conduct product development and processing for preservation. I think we have a particular strength in food chemistry and nutritional biochemistry in this program. preclinical screening and mechanistic studies, especially in nutrition medical labs where using animal models and cellular systems. We're fortunate that we can conduct phase 1, phase 2 clinical trials right here on campus. A faculty member, Neil Hooker, in the Glenn School, interested in marketing, regulatory, and agricultural economic studies. And another arm of this program deals with entrepreneurship and technology transfer. So as you heard earlier this morning, Ohio State University is really quite unique. We're a comprehensive institution. We're one of the few institutions that have both a College of Medicine and a College of Agriculture, Food, Ag, Environmental Sciences within the same campus boundaries. In our medical center, 3 university hospitals, a clinical research center that helps us quite a bit in this program, a heart hospital, general medical center, and the James Cancer Hospital and Solov Research Institute, where we just have a world-class comprehensive cancer center. And my good colleague, Steve Clinton, who you'll hear from in just a few minutes, leads the molecular carcinogenesis and chemo prevention program. And we also have, and it looks like the slide's cut off here, this a unique shared resource. This was a partnership between our college at OARDC and the Comprehensive Cancer Center that established this nutrient and phytochemical analytic shared resource, which helps investigators with their analytical needs for research in this area. This program has received attention by the agricultural community in Ohio. This Growing the Cure campaign was started by 5 agricultural cooperatives. And they've come up with these really nice slogans: food fights cancer, growing the cure. Agricultural Secretary Tom Vilsack came. You can see here over to our laboratories. And this campaign was organized and kicked off at the last Farm Science Review. So much of our work in the center was sort of guided by epidemiology early on. We heard quite a bit about some of the limitations of epidemiological type studies. But there was this really nice review conducted back in 1996 by Steinmetz and Potter on vegetables, fruit, and cancer prevention. And what these authors did is they reviewed more than 200 different epidemiological studies in the literature. And from those 200 studies, they found that the types of vegetables or fruit that most often appear to be protective against cancer are raw vegetables, allium vegetables, carrots, green vegetables, cruciferous vegetables, and tomatoes were also highlighted. And I'll be talking a little bit more about tomatoes, of course. The bioactive phytochemicals that we've been monitoring in our program include the carotenoid compounds, and I'll be focusing just to tell you a little bit about a few of our studies there. The isoflavones in soy, glucosinolates and isothiocyanates in cruciferous vegetables, and flavonoids and phenolics in small fruits and vegetables. And Steve Clinton will be talking more about the transdisciplinary work that we conduct in the center on different food products that have been developed and targeted towards cancer prevention. So my particular interest on some of these phytochemicals deals with the carotenoid compounds. I want to give you a little overview and discuss a few short studies about these compounds in the diet. I think everyone here knows they're responsible for the red, yellows, and orange colorations in fruits and vegetables. There's about 700 different of these compounds that have been identified. One thing you should keep in mind is that primarily plants and photosynthetic microorganisms biosynthesize carotenoids, but animals and humans must derive their carotenoids by consumption of plant products. The biological functions are numerous. Of course, in nature, they're very well known as secondary pigments in photosynthesis. They serve as a photoprotective agent from the damaging effects of light on plants, but also in humans. In the back of our eye, there's two carotenoids known as lutein and zeaxanthin in the macula pigment. They're also photoprotective for those individuals that have a decreased density or low pigment concentration of these carotenoids in the back of our eye. They're at a higher risk for age-related macular degeneration, one of the leading causes of blindness in the elderly. One of the well-known carotenoid is beta-carotene, has the highest pro-vitamin A activity, so it can be cleaved in the intestine and the liver to vitamin A. Non-pro-vitamin A functions of these compounds include singlet oxygen quenching, probably the most efficient singlet oxygen quenchers of any naturally known compound. Antioxidant activity. When fed to animals, they can enhance the immune response, and there's more and more evidence that these compounds in the diet may have potential chemopreventative properties. In fact, one of the first suggestions that beta-carotene, dietary sources of beta-carotene, might be associated with a decreased risk of cancer was published by Pito, Dahl, Buckley, and Sporn in Nature back in 1981. I was a graduate student at the time, and this really spurred a lot of activity on carotenoid research as potential chemopreventatives in the diet. This was one of the stronger epidemiological studies that was published in the literature. It was a seminal article by Ed Giovannucci and co-workers at the Harvard School of Public Health. And it was on the intake of carotenoids and retinol in relation to the risk of prostate cancer. And what Dr. Giovannucci and his group did is that as part of the health professionals follow-up study, it was a relatively large study, 40,000 men were enrolled in this study. And what they did is they followed the diets of these men for more than a 10-year period. And of all the fruits and vegetables that they consumed in the diet, it was the combined intake of tomatoes, tomato sauce, and pizza, accounting for about 82% of the red pigment in tomatoes, lycopene, was inversely associated with the risk of prostate cancer. And we being involved and interested in carotenoid research, this was quite exciting at the time. And we became very interested in studying these compounds. And this is some data actually from Dr. Giovannucci's publication on this relative risk of prostate cancer and the intake of tomato sauce in the health professionals follow-up study. And you can see here this very nice trend, but for those individuals, men that consumed greater than 5 servings of tomato sauce per day in their diet had about a 30 to 35% decreased risk of prostate cancer, and it was a highly significant trend. So we became quite interested in the carotenoid pigments in various tomatoes. We've been working with Dave Francis in horticulture and crop science at OARDC. He's been providing us some unique varieties of tomatoes. The tomato on the right here is a high lycopene variety, and it contains lycopene in about 2 to 3 times the typical concentrations that you might find of the tomatoes that you can buy in the supermarket. The type of lycopene that is biosynthesized in this tissue is this all-trans configuration. For those of you who can see the molecular structure, so it's a linear type molecule. This tomato on the left also contains lycopene. It's called a tangerine variety. And it's unique in that the biosynthesis of lycopene in this tissue occurs in this bent form, this tetra-cis form it's known. What's interesting is one of my PhD students just completed a bioavailability study following the absorption of lycopene from from a red versus a tangerine variety, and found that actually lycopene in this tangerine variety is about 12 times more efficiently absorbed during the digestive process into the bloodstream relative to these red varieties of tomatoes. And we hypothesized that one of the reasons that this occurs is because of the way the carotenoids are actually biosynthesized and compartmentalized in the tissue. Using the Molecular and Cellular Imaging Center at OARDC, Jessica found that the tangerine tomato, the lycopene is actually compartmentalized in the plastoglobuli in small lipid sort of droplets, while in red tomatoes you can see crystals actually of lycopene because of the structural configuration. So these matrix effects have a huge influence on the absorption characteristic and the bioavailability of this particular phytochemical. And I think Henry had alluded earlier on about a number of questions we have about bioavailability and how can we enhance absorption characteristics of these important phytochemicals into the bloodstream and to the target tissues. Well, I'm sorry this is a little cut off here, but this is one of our clinical study designs that we conducted with Steve Clinton over in the Comprehensive Cancer Study. And we've been doing bioavailability studies on tomato products and lycopene-containing products on healthy individuals for many years. But this one is with prostate cancer subjects. Small clinical pilot trial, about 32 subjects enrolled. Usually we place people like this on a washout phase, lycopene-free diet for 7 days, and then they were randomized to one of 4 nutritional interventions, consuming a vegetable juice, a tomato soup, or a tomato sauce. If you're familiar with these brands, you would know who helped support this study. And we adjusted the serving sizes that these men consumed so that they consumed about 20 milligrams of lycopene from these products each day, and a control group that consumed a soy protein isoflavone-based beverage for a 3-week period, and then they were scheduled for their surgeries and had their prostates removed. So the nice thing about this pre-surgical design is that we could obtain blood samples throughout the study, but also biopsy samples of prostate tissue for our analysis. And those of you who know what I like to do, we developed some really nice methods to measure these compounds. We followed the change in the plasma lycopene concentrations in the bloodstream, significantly enhanced from the 3 nutritional interventions, as you see here. The control group, a significant drop. So that was sort of expected, this plasma response. More interestingly was what did we actually deliver to the target tissue, in this case to the prostate gland. Unfortunately, we only had small samples of biopsy tissue to work with, so we had to develop some highly selective and sensitive methods using HPLC tandem mass spectrometry. And we were able to very selectively monitor all the lycopene and the different cis isomers and beta-carotene deposited in the prostate tissue. And in the control group, we could see quite nicely these soy isoflavones that were also deposited in the target tissue. These are weakly estrogenic compounds. Genistein and diazine also have an interest in prostate cancer prevention with these soy isoflavones. So here you see just some data. I don't have too many data slides here, but I just wanted to share this one with you on the lycopene deposition after this 3-week intervention. And if you just look at this total column here, you'll note that the 3 intervention groups, the sauce, the soup, and the juice, they all had a significantly higher deposition of lycopene in comparison to the control group, that on the soy-based protein isoflavone beverage. And I think what's important about this particular observation was that not only can we just increase the plasma levels, which we expected, but we could actually significantly alter the concentration of lycopene in the target tissue within a short nutritional intervention. It only takes 3 weeks of consuming these products to significantly enhance the level of these compounds in the prostate that are associated with prostate cancer. We know that in order for carotenoids to be efficiently absorbed, and we're very interested in how can we enhance the bioavailability and the absorption into the bloodstream, that they're first micellarized during digestion and taken up into the intestinal enterocyte. This is where they can be metabolized to vitamin A, like beta-carotene or other provitamin A carotenoids. But since these compounds are highly lipophilic, they're carried on this chylomicron lipoprotein fraction that's secreted in the lymph for transport in the bloodstream. And what's nice about this is that we could obtain a measure of newly absorbed carotenoids if we measure those that are coming through the intestinal wall on the chylomicron fraction. And then we could study what's the effect of food matrix or food formulation on the absorption of carotenoids from a meal or different meal formulations. So I want to share with you just some data from a small pilot clinical trial we did on absorption of carotenoids and the influence of food formulation. This is actually data taken from individuals that came into the clinic, and for breakfast early morning we fed them a large vegetable salad and tomato meal. And we put a catheter in their arm and we take blood samples all day long, 9-10 hours you see here. And we measure the carotenoids on the chylomicron fraction. So this is what's coming through from that breakfast meal. You can see at time 0 there's hardly any absorption. It takes about 4 to 5 hours to see the maximum absorption of alpha-carotene, beta-carotene, and lycopene after we consume a meal. And then it drops off throughout the day, and you just sort of measure the area under the curve, and that's known as the postprandial response. Well, what was an interesting observation was that when we fed these individuals, and these were students, by the way, when we fed this vegetable salad and tomato meal with a no-fat salad dressing, there was hardly any response. And when it was fed with a reduced fat salad dressing, there was sort of an intermediary response. And with a full fat dressing was the response that you see on this slide. And here you see some of this data. The greatest response was with the full fat dressing for alpha-carotene, beta-carotene, and lycopene. This intermediary response with reduced fat dressing. And really quite surprisingly, hardly any absorption of these carotenoids when there's a fat-free salad dressing, confirming that the co-consumption of fat is absolutely essential for the efficient absorption of these carotenoid compounds into the bloodstream. So we continued some of this work with our interest in seeing how lipid modulates bioavailability of carotenoids. This is another tomato variety that Dave Francis at Horticulture provided to us, a high beta-carotene variety of tomato. A lot of interest in these high beta-carotene crops in the biofortification arena to be used in different areas of the world where vitamin A deficiencies are still prevalent. And one of my students, Rachel Kopek, monitored the absorption of these carotenoids, specifically beta-carotene from this novel tomato. Actually first processed the tomato into tomato sauce over in the food science department when it was co-consumed with and without lipid. But in this case, we used avocados as the source of lipid, where avocados contain about 17% lipid in that fruit. But we also monitored the bioconversion of beta-carotene to vitamin A, again influences the presence of lipid. And what Rachel found was that As expected, there was an enhancement in the absorption of beta-carotene in the bloodstream when the avocados were co-consumed as the source of lipid, about a 2.4-fold enhancement. But also the metabolism to vitamin A, retinol palmitate, delivered a 4.6-fold enhancement in the delivery of vitamin A into the bloodstream when this tomato was co-consumed with dietary lipid. So this message has been picked up in the marketplace. You can see this picture here of Wishbone's salad dressing. They've put a consumer message on their label from these types of studies that now says naturally helps better absorb vitamins from salad with the oils of Wishbone. So this is a case where we were able to translate some of this information out into the marketplace. So let me just summarize then a few highlights. That the CAFRE Crops to the Clinic program really explores kind of a unique area that's focused on functional foods for health, particular emphasis on cancer prevention, given our collaboration with the Comprehensive Cancer Center. I think just a wonderful opportunity enhances collaborations among investigators, especially horticulture, food science, nutrition, and medicine. And for students and researchers in the laboratory, it provides just a fantastic opportunity to learn and train in this multidisciplinary environment. And our students move from laboratory to laboratory, and their experience, I think, is just amazingly enriched. We've been able to successfully leverage some internal seed funding from OARDC and the Comprehensive Cancer Center, and I believe that this really makes us much more competitive for external dollars from federal agencies, and that this interdisciplinary program, I think, fits well with our new discovery themes that are coming, especially health and wellness and food production and security, and look forward to expanding some opportunities there. Of course, these are just a few of the individuals that have been quite involved in our research program. There's about more than 35 faculty somewhere involved in the CAFRE program on campus, and to my graduate students and postdoctoral researchers who are responsible for generating much of the data that I shared with you today. Also, the generous support for this program, I mentioned OARDC, the Medical Center, Comprehensive Cancer Center, federal agencies, some of our recent industry partners, and the donors such as the Growing the Cure campaign. And I think most of you here know on campus our Pelotonia ride. Within the Pelotonia, we have a Crops to Clinic team. Please join us on our ride. Here's a photograph from some of us from last year's Pelotonia team. Robin Ralston, back over here. She's our co-captain with me this year for the 2013 Crops to the Clinic team. And she informed me that you don't have to ride. You can be a virtual rider and still join our team and support this effort for funds for cancer research. So with that, thank you very much.